Hi. So, before we move forward with today's discussion, I need to give you a quick disclaimer, which is that typically once one looks at the topic of genetic drift, it is typically followed by Moto Kimura's neutral theory. However, in the context of the present course, I am moving the neutral theory to week 10, when we are going to discuss about how genomes evolve. Primarily because this week there are many other things which need to be covered in some amount of detail, and B, generally I have seen that this entire neutral theory thing makes a lot more sense when you study it in the context of genome evolution. So, that is one quick departure that we are making from the announced syllabus. So, in the last discussion we talked about genetic drift, and we said that there are other evolutionary forces which we need to look at now, and the one that we are going to look at during today's discussion is mating. So, if you remember, one of the conditions of Hardy-Weinberg equilibrium is that mating needs to be random. Now, what do we mean by that? So, random mating, or also known as panmixia, by definition is when any individual of one sex has equal probability of mating with any individual of the other sex. So, obviously this allows us to do those nice Punnett squares and lead to go from allele frequency in the parent generation to the genotypic frequency in the next generation. However, as we all know, in real life much of the time mating is not really random, and therefore non-random mating is simply any departure from the above condition, and there are two major ways in which mating can be non-random. One is what is known as assortative mating, where individuals who have similar phenotypes, they mate preferentially with each other with a greater probability, and the other is what is known as disassortative mating, where individuals with dissimilar phenotypes, they tend to mate with each other with a greater probability. So, we will consider each one of them in turn and see what they end up doing for the evolution of the population. So, if you look at assortative mating, so there are many examples. So, for example, one of the most well known thing amongst humans is that in many societies, particularly Western societies, it has been shown that people tend to have assortative mating for height. In other words, tall males typically tend to mate with tall females and vice versa. However, the point to be noted is that many a times assortative mating can happen even if there is no direct mating preference. So, that is one example that I would like to give you over here. So, this is an example of this plant called insect called Echinopa. This is a kind of tree hoppers, and these guys, they feed and develop on multiple plants. Now, turns out that those that feed on the similar plants or the same plant, they tend to mate with each other much more frequently. So, one can obviously say that this is an assortative mating for feeding on the same plant. But if you look at it a little more closely, as was done by Wood and Keyes in 1990, they figured out that that is not exactly what is happening. So, turns out that the chemistry of these plants differ slightly, and that determines what is the rate at which the insects which are feeding on them are going to develop. Now, when the insects, the males and the females, they are feeding on the same plant, they tend to develop at a similar rate, and therefore, they become sexually mature at the same time. Now, turns out that in this particular species, the females are sexually receptive for a very small amount of time. So, obviously, the only males that are available to them during that small duration are the ones which have essentially co-developed with them at roughly a similar rate, and those happen to be the ones which are on the same plant. So, Wood and Keyes showed this very nicely by providing the females with sexually mature males which were actually developed on different hosts. So, they artificially provided these males and ended up showing that this entire preference for mating for with males from the same plants, that completely disappears when there is availability of other kinds of males. So, this is just an example to show that not all assortative mating is necessarily through mating preference. Similar stuff has been shown in Drosophila. So, for example, if you have Drosophila which are coming out of a crowded cultures, there is assortative mating. So, the development time of Drosophila becomes longer when you have a larval crowding, and all the flies who come out early, they tend to mate with each other. All the flies who come out late, they tend to mate with each other. But this is not really because there is any major preference. It is just that when the females are sexually mature, the only males that they find are males who are of the early mating type or males who are of the late mating type, depending on when the females have come out. So, this is a reasonably well known thing that not all assortative mating is due to mating preferences alone. So, what exactly does assortative mating lead to? In order to understand that, we will do a quick example, and in this case we will take an extreme case of what is known as complete assortative mating. So, let us assume that we have a one locus two allele scenario, and let us assume that our two alleles are A1 and A2. So, that we have A1A1, A1A2 and A2A2, three genotypes. Let us also assume that these three genotypes have three different phenotypes, such that the phenotypes are visible, the phenotypes are something that the genotypes can distinguish, and let us assume that A1A1 phenotype only mates with A1A1 phenotype, A1A2 phenotype with itself, and A2A2 similarly mates with A2A2 phenotype. So, in some sense this is like selfing, except that in selfing the individual mates with itself. In this particular case, the individual is mating with individuals of its own genotype, that is all. Now, suppose we have a scenario like this. What is going to happen? You are going to get three kinds of individuals in the next generation again, A1A1, A1A2, A2A2. However, the way they will be produced will be slightly different. So, all the A1A1 individuals with probability 1, they will produce A1A1 offspring. Similarly, all the A2A2 individuals with probability 1 will produce A2A2 offspring, right. What will happen to the heterozygotes? A1A2 heterozygotes, when they are mating with each other, then they are going to produce all the three genotypes A1A1, A1A2, A2A2, and assuming all other Hardy-Weinberg laws to be Hardy-Weinberg conditions to be operative, these will be produced in proportions 1/4 A1A1, half A1A2 and 1/4 A2A2, right. So, if that be the case, look at what is happening to the A1A2 genotype or phenotype, whatever. What is happening is that they are producing babies, offspring, of which 1/4 are of the A1A1 phenotype, 1/4 are of the A2A2 phenotype, and only half are the corresponding phenotype, which is A1A2, right. So, in other words, the A1A1 and the A2A2 are only producing babies of their own type, but A1A2 is producing only 50% babies which are of its own type. The remaining 50% are going and getting added to the A1A1 and the A2A2 phenotype. So, what happens if this continues across generations? I mean, you do not even need to do any maths. You realize that if each generation 50% of the offspring are being lost to the other phenotypes, then after some time all the A1A2 genotype in the population will cease to be, and the population will now consist only of A1A1 individuals and A2A2 individuals, right. So, just to show you what is happening, if you look at the next generation, the P prime. So, let us assume that capital P, capital Q and capital R, these are the three genotypic frequencies for A1A1, A1A2 and A2A2. So, obviously, since 50 and 1/4 are coming over here. So, P prime will become capital P plus 1/4 Q. Similarly, R prime will becomes capital R plus 1/4 Q. So, the 1/4 is coming from here, and this Q prime, capital Q prime, is going to become equal to half of Q, right. As I said, in the long run, q has to become equal to 0. But what will happen in the next generation? So, in the next generation, suppose we are trying to track the allele frequency small p, what will be small p? Remember, small p is always equal to capital P plus half q. Therefore, allele frequency small p prime in the next generation is going to be equal to capital P prime plus half q prime, right. Now, we have already given the expression for capital P prime and capital Q prime. So, you simply add those up. So, P plus 1 by 4 Q, this is capital P prime, and this half into half Q, this is capital Q prime. So, this is 1 by 4 Q, this is 1 by 4 Q, add the 2 up, it becomes half Q. So, this becomes equal to capital P plus half Q. But as we already know, capital P plus half Q is simply equal to P, which is the allele frequency of allele A1, right. In other words, in the next generation, the allele frequency is not really changing in the population. All that is happening is that the genotypic frequency ends up changing. I mean, this you can also see intuitively, right, because there is no selection happening, no allele is getting lost, no allele is getting created. All that is happening is that the alleles are getting repartitioned in amongst the genotypes in a particular way. Therefore, the allele frequency does not really change. What changes are the genotypic frequencies, and that is what I am trying to show you, tell you over here. Because the allele frequency is not changing, therefore, if you are defining evolution as change in allele frequency, there is no evolution happening at the level of that locus. However, if you are defining evolution as change in phenotypic frequencies, then you can clearly see that when you are starting with 3 different phenotypes, the proportions of those phenotypes are changing, and after some time you are left with only 2 phenotypes, right. And not only are you left with 2 phenotypes, you are left with the corresponding homozygotes, right. It is the heterozygote which disappears, and it is only the homozygotes that will survive in a condition like this. So, this increase in homozygosity, this is the main point that we need to take from this. So, here I am showing it to you in the context of strict selfing. However, if you have assortative mating of any kind, typically this is what ends up happening. The homozygosity, the proportion of homozygotes in the population keeps on increasing, and the heterozygosity, the proportion of heterozygotes in the population keeps on going down. Now, what is the net result of that? Now, suppose you have a population in which you have many, let us say, rare recessive alleles. Okay. Now, most of the diseased alleles that we have, those are actually of the recessive type, not all but many. Now, when they are in low frequency, remember those alleles are going to be found in the heterozygotes, and therefore, they are not going to be seen by selection because they are not getting expressed. However, when you have the homozygosity of the population increasing, then many of these rare alleles are now going to get expressed simply because they are now coming in as homozygotes in the genotypic sense. And when that happens, all these rare alleles, the diseases caused by them, their frequency starts going up in the population, and as a result the overall fitness of the population starts suffering. So, just to give you one example from humans. So, there is this thing known as early onset deafness in humans, which basically means from a very young age people go deaf. Now, this can happen due to multiple reasons. In many cases, it is related to diseases and accidents. However, in about 68% of the cases, this is purely genetic, and people have looked into it. This is not caused by a single gene. There are about 115 genes and mitochondrial DNA variants. Most of them are autosomal and recessive and very rare, except one. Which one? So, there is this gene called the GJB2, which codes for a gap junction protein called connection 26. Now, turns out that if you have a particular mutation called 35 del G mutation, basically a 35 base pair mutation at this locus, then in the homozygous form this can lead to early onset deafness. Now, turns out that the frequency of this particular allele in the population in US and Europe is about 0.01, which is 10 to the power minus 2. So, if that be the case, what is my expected frequency of the homozygotes? So, this frequency is 10 to the power minus 2. So, expected frequency assuming Hardy-Weinberg is going to be square of that, right. In other words, the expected frequency under Hardy-Weinberg should be 10 to the power minus 2 into 10 to the power minus 2, which is 10 to the power minus 4, which is about 1 in 10,000. However, if you actually look at what the frequency is, you find that it is about 3 to 5 times greater than that, which means it is about 3 to 5 in 10,000. So, what exactly is happening? Why is this, I mean, this is statistically significant. So, why is it statistically significantly larger by about 3 to 5 times? So, turns out that very often, since this is early onset of deafness, kids who have this disease, they end up going into special schools for people with hearing impairment, and there they meet other people who also have the same problem. Now, because they are learning together, many a times obviously they learn sign languages and other things, due to which they are able to communicate with each other. And of course, they are able to communicate with each other a lot better than with people who either do not know the sign language, or with people who let us say have learned it but are not very proficient at it. Therefore, it is known that there is a very high degree of assortative mating between people who are deaf. In the US the number is about 80%, more than 92% in Europe, and once you factor that into your models, you realize that the observed frequency matches the expected frequency very well. So, essentially the reason that this particular recessive homozygous recessive allele is in high frequency, in the genotype is in high frequency in the population, is simply because of assortative mating. So, there are a few general insights about assortative mating that we should keep in our mind. One, as I said, that it increases the frequency of homozygotes at the expense of the heterozygotes. More importantly, if you have assortative mating, then the population can become split. Okay, it can become divided into multiple subsets due to genetic differentiation. So, this is something to which we will come back again when we do our speciation topic somewhere in the future. Okay, at that point we will talk about this again, but right now just keep a note somewhere that assortative mating, because it can lead to genetic differentiation and substructuring within a population, it is a very potent force for microevolution, particularly speciation, formation of new species. Okay. So, of course, that was about assortative mating. Now, the other kind of non-random mating, as I said, is disassortative mating, where you have mating of individuals with dissimilar phenotypes. So, this is a little less common than assortative mating. However, there are a few very well known examples, and one of the well known examples is this famous Wedekind et al. Experiment in 1995. So, what Wedekind et al. Did was they recruited a bunch of college students, males, and then requested them to wear the same t-shirt for about 2 days. And then they took all kinds of precautions. They were to make sure that there is no other odor that comes into the t-shirt. And after 2 days, they took those t-shirts, they put them in plastic bags, and then they recruited a bunch of females and told them to smell those t-shirts and rate the smell in terms of attractiveness and nothing else. And on the sides, what they also did was they looked at the kind of MHC gene. Now, you must have heard about the MHC gene, major histocompatibility genes. So, these are genes which are very important for the immune function of humans, and therefore, this is a very highly variable kind of a gene. So, when they did the profiling of both the males and the females, and they tried to correlate the MHC profile of the females with what kind of males they are choosing, or what kind of males they are saying is they are finding attractive based on the smell alone, they found that females typically tended to choose males who had a different MHC profile compared to those who had same MHC profile. In other words, if the male and the male chose those, sorry, the female chose those males who had the opposite MHC profile compared to the ones who had the same MHC profile as the female concerned. So, this is a very well known experiment, and similar stuff has been repeated in terms of mice. Again, similar results have been shown. The similar thing has been done in fowl, jungle fowl. There they inverted it. There they asked if the males, what kind of females they end up choosing, and very interestingly turns out that there was no major choice in terms of mating per se, but there was a difference in terms of how much the males are investing in terms of their sperm output. So, turns out when the females hens had a different MHC profile, the males tended to have a greater sperm investment compared to when the females had the same MHC profile as the males. So, this is one very nice example of how the MHC, major histocompatibility complex genes, they end up affecting our perception of how desirable or how attractive the opposite sex is, or individuals of the opposite sex is. Now, I will not get into the models and everything, but generally this assortative mating leads to an excess of heterozygotes with respect to the corresponding Hardy-Weinberg predictions, and as a result of that, exactly the opposite of assortative, it ends up maintaining polymorphism in the population, and therefore it prevents the population from subdividing. So, those were about two departures from random mating. There is one more thing which is very important in this context, which is inbreeding. Now, what is inbreeding? Inbreeding is mating between individuals that are genetically closely related. Now, please appreciate one thing over here. When we define random mating as all individuals of one sex have equal probability of mating with individuals of another sex, that also refers to close relatives and everybody else in the population. So, even under random mating, you expect some amount of inbreeding to be happening. Okay. Here, when we are talking about inbreeding, we are essentially talking about when that is happening in a much bigger way, mating between genetically close relatives, and turns out that like assortative mating, this also ends up increasing homozygosity. Now, as I said, anything that increases homozygosity, that is going to increase the probability of recessive deleterious alleles coming together, which in turn can have negative consequences at the population level. Now, this has actually led to a term known as inbreeding depression, which is the reduced survival and fertility of offspring of individuals who are related to each other. Now, you can see a pattern over here, right? Genetic drift leads to homozygosity, assortative mating leads to homozygosity, inbreeding also leads to homozygosity. Therefore, although the term inbreeding depression is typically named in the context of inbreeding, it is essentially talking about something, increased homozygosity, which is common to multiple evolutionary forces. That is why sometimes instead of saying inbreeding, this entire thing about homozygosity leading to a reduction in fitness, it is actually called inbreeding like effects. Okay, that is a better way of saying it, but sometimes they simply use the term inbreeding depression, but inbreeding depression does not necessarily need to come only from inbreeding, it can be coming from, as I said, genetic drift or assortative mating, right. So, what exactly can inbreeding depression lead to? As I said, bad things. How bad? Let me give you 2 or 3 examples. So, when it comes to inbreeding, the textbook example is this unfortunate gentleman, King Charles II of Spain. So, King Charles II belonged to the famous Habsburg family of Austria, and it is one of the most powerful royal families of Europe. Okay. So, it ruled the Holy Roman Empire for about 350 plus years, and this family produced kings for dozens of countries and empires, right. So, it was a huge family, lots of branches, but one of the things that they did very consciously was that they heavily intermarried within the family. So, first cousins, second cousins, lots of marriage happened, primarily for political reasons, to make alliances, to make sure that king of A does not launch an expedition against king of B and so on. However, this massive amount of inbreeding led to very major physiological problems in the Habsburg family, and probably the biggest example of that is this gentleman, King Charles II. So, as it says, he lived between 1661 to 1700. He became king at the age of 4. However, right from childhood, it was clear that the kid was suffering. So, he had this, you can see his jaw, right? Can you see how it is protruding below like this? That is the famous Habsburg jaw. So, this is a condition where the jaw grows like this, elongated, and because of this it is extremely painful. He could not chew. He had a very long tongue, because of which he could not speak properly. He had speech issues, and people around him could not understand. But more importantly, he had very severe cognitive issues. He could not speak, he could not walk till the age of 8, and even then he just could not learn. And this guy was like the emperor of Spain and one of the most powerful people in the whole world. So, because he had cognitive issues, he was not even educated, and most importantly in this particular context, he was also infertile. He could not produce a baby, and because of which the entire Habsburg dynasty in Spain ends with this particular person. Basically, there was a massive war after he died because he did not have any successors, and essentially the Habsburg line in Spain ends with this person. Now, this particular case of what inbreeding can do, and particularly what it did in the Habsburg family, is a fascinating case, lots of information, very well studied. So, I will guide you towards these two videos, excellently done videos. Although they are talking a lot about the history, but they are also talking a bit about the genetics. So, have a look at it, and as usual, the contents of these videos are for fun, they have nothing to do with the exams, but that should not prevent you from looking at the videos anyway. Now, in an ideal world, I should have been able to tell you that it is a disease, inbreeding is a disease of the royals alone and normal people are not affected by it, but unfortunately that is not correct. So, turns out that this thing about marrying relatives, it is actually there in many parts of the world, and wherever it is there, it typically leads to lots of problems. So, here I am showing you some data from a paper by Beatles et al. In 1994. So, what they did was they went to 38 human populations from all over the world, and for each population they looked at some marriages which have happened due to mating between first cousins, and they also looked at those marriages which have happened because of mating between unrelated couple. Basically, the male and the female are not related to each other, and for both sets they tried to figure out what is the average percentage of mortality in the babies which are arising out of those marriages. So, here what you have is the mortality of children of unrelated parents on x axis and the percentage mortality of children of the first cousins on y axis. And if you look at this red line is assuming that the two mortalities are the same. Okay. And basically what you see is that in all the cases the mortality, percent mortality of the first cousin kids coming out of first cousin marriages is much greater than, I mean, okay, this is one little exception, but otherwise it is mostly much greater compared to the mortality which is coming out of children coming out of unrelated parents. So, this is a very stark example of what inbreeding depression can do, and this is just, we are talking about one generation stuff over here. This is not even a multi generation stuff, okay, in one generation what it can do. To give you an example in wild animals, so this is the famous Soay sheep, extremely well studied. In this particular case, we are talking about a breed of sheep on the island of Hirta near Scotland, okay. So, this is a population that was established in 1932 with about 107 individuals. So, not a very small founding population, not very large either, somewhere in the middle. And this population size, it fluctuates between about 500 to 2000 sheep. It is an island, right. So, depends on all kinds of environmental conditions, and because of this fluctuation, it has undergone a lot of drift, and that drift, as I told you, is showing up as inbreeding depression. So, what Coltman et al. Did was, they took a large number of these sheep, and for each one of them they did a lot of sequencing to figure out that what is the fraction of heterozygosity within the individuals of the sheep, in that population. So, this is heterozygosity measured at the individual level, and then they grouped it in 4 equally spaced groups. So, if you are into statistics, basically the 4 quartiles, and that is what I am calling here low, medium, high and high, okay. That is the heterozygosity levels. And for each one of them, in each one of these groups, they tried to see what is the survival probability, and that survival probability is what is being plotted over here. So, as you can see that as you increase the heterozygosity levels, the predicted survival probability increases almost linearly. I mean, that line is the regression line. You can see that it is very close to linear. Now, what survival probability is this? This is the survival probability of the lambs during the winter. So, winter is bad in Scotland, right. So, they are trying to see that what is the probability that the lamb will survive, and how does that probability change with heterozygosity, and as I said, it increases linearly. Now, why does it happen? What exactly is heterozygosity doing over here? So, the reason I have chosen this example is because here they actually figured out what is happening. So, turns out that the individuals who have greater heterozygosity, they have greater tolerance towards parasites of the guts, specifically nematode parasites, worms of the gut, okay. Now, that is what is leading to the greater survivorship with greater heterozygosity. Now, if this is indeed correct, then what do you expect? You expect that this is happening because of the nematodes. So, if you remove the nematodes, this pattern will disappear, right. That is exactly what they ended up seeing. So, what they did was they treated a group of these individuals with antihelminthic drugs. So, antihelminthic drugs will take out all the worms, and the moment that happens, this survivorship differences based on heterozygosity, that completely disappears. So, this is what tells you that in this particular case, the heterozygosity is operating through the presence of the nematodes, right. Okay. So, a quick recap, quick retrospection. Okay. We have till this point studied 4 or 5 different evolutionary forces. So, we looked at selection, and we know that selection ends up reducing the variation in a population, and in terms of its strength, it can be a very strong force. I mean, depending on the selective selection differential, depending on how much variation is there, etc. But in the correct condition, it can be a very strong force. We looked at mutation. We said that it is one of the major ways in which the variation in a population goes up, but we also saw that it is in general a very weak force. It does not change the composition of the population extremely fast, and particularly when it is the population is small, when it has to interact with drift, then it becomes an even weaker force. Then we looked at drift itself, and we saw that drift in general reduces variation because there is loss or fixation of an allele. It also reduces homozygosity, and anything that reduces homozygosity, as we saw, can reduce the overall fitness, and because of the reduction in fitness, selection will become important, and therefore through an indirect effect it can again lead to a reduction in variation. And as we saw that it can be strong in very small populations, but otherwise generally it is weaker than selection. In large to moderately large populations, it is a weaker force. Then we looked at assortative mating in today's discussion, and we saw that it reduces fitness through reduced homozygosity, and again, as I said, anything that reduces fitness will typically end up having a negative effect on how much variation is there, and sometimes assortative mating can be a strong evolutionary force, as I told you. And finally, we looked at inbreeding, which again reduces fitness through reduced homozygosity and therefore can reduce variation. And again, as I told you, inbreeding in many cases, particularly when population sizes are small, can be a real big issue. So, what is this table telling us? This table is telling us that most evolutionary forces, barring mutation, they will typically lead to a loss of variation either directly or indirectly through the fitness effect, as I talked about. Now, if this be the case, the only force which is opposing it, creating new variation, that as we saw is a weak force. Everything else is a pretty strong force either by itself or under the correct condition. So, if that be the case, then taking point 1 and point 2 together, most variation should have disappeared by now. Okay, we know that mutation rates are typically not very high. We also know, as I said, it is a weak force. So, all the opposing forces are strong, all the creating force is weak. So, most variation should have disappeared by now. But is that the situation? Remember our first discussion where we talked about the alien. One of the things that our alien saw was that no, there is a lot of variation. Okay, that is what kind of makes life so interesting to study. So, if that be the case, where exactly is this variation coming from? There has to be some evolutionary force which is allowing the populations to regain the variation, to retain the variation, at least to some extent. Okay, it cannot be one little mutation acting against all these other forces put together. What is that evolutionary force? That is what we are going to look at in our next discussion. See you. Bye.